Thanks for having me here. Hi, so today I'll be going over my project that is identifying associations of rare non-coding variants with autism spectrum disorders through integration of gene expression, sequence, and sex information. So de novo variants are a class of mutations that are only being found in the children but not in the parents. They're extremely useful to study these sort of complex disorders because if the parents don't have it and the child has it, you can directly identify that these variants may or may not have some higher impact on the disease. So previous studies related to these de novo variants are mainly restricted to the coding regions through these exome sequencing studies. And these studies have extensively identified quite a lot of risk genes implicated by de novo coding variants in ASD. But on the other hand, the contributions of de novo variants in the non-coding region remain less clear despite some early evidences through whole genome sequencing studies. Here is one of the whole genome sequencing studies that is related to the de novo variants in autism. In this study proposed by the Ogun Troy Squire group from Princeton, they used a deep learning-based method to analyze the contributions of de novo non-coding variants. They utilized this Safari Simon simplex collection data set, which contains thousands of simplex families with one proband with autism and a sibling unaffected. So the authors of this manuscript employed this deep learning-based framework. First of all, the input is the 201 flanking sequence surrounding the variant, with the mutation being in the center. And then they used their previously proposed deep learning network, that is the DeepC, to predict the chromatin effects such as histone modifications, transcription factor binding sites, and DNA's hypersensitivity sites from the 200 base pair sequence. And then they train a binary classifier that learns to distinguish between the pathogenic and benign variants from the predicted chromatin effects. And this classifier outputs a singular disease impact score, which quantifies the pathogenicity of the variant with respect to the disease. And after applying this entire deep learning framework to the de novo variants in the Simon simplex collection cohort, the authors, one of the main findings is that they observed this significant difference between the proband and sibling de novo variants in terms of the DIS score. And such difference is mainly concentrated in the variants that are close to the genes that are being overexpressed in the GTEx brain tissues, as shown here. You can see that all these red dots are the variants that are the GTEx brain tissues. Now, from this observation, it's seemingly clear that the deep learning-based model is having some predictive power when it comes to autism pathogenicity of the de novo variants. But then again, here in my talk, we will try to gain a better understanding of what's actually going on with this, which is presumably deep learning-based signal, and I will propose an alternative analytical approach to further elucidate this signal. So one of our major observations by interrogating this deep learning-based score is that we observed a very strong or even almost monotone relationship between the deep learning-based score as shown in the y-axis and the local GC content surrounding the variant, which is showing the x-axis. Here, the local GC content is defined as just the total number of GNC nucleotides in the 200 base pair flanking region of the variant. And as you can see, there's a very high Spearman correlation. And if you inspect the mean of the DIS score at each of these evenly spaced local GC content bins, you will see that the relationship between the mean and the DIS is almost monotone. Now, this raises some concerns, and of course, this question, that is, now that the authors have already used the deep learning-based DIS score to produce so many ASD association findings, can we produce exactly the same findings, but using local GC content, which is a simpler sequence metric, as opposed to using the DIS? Here, we try to replicate the author's analysis, but using the local GC content in place of the deep learning-based score. These are exactly the same points as shown in the previous GTEx tissue-based graph. Each of these points is a GTEx tissue, and the x and y axis represent the male versus the proband versus sibling differences in local GC content and DIS score respectively. And you can see that using the local GC content alone, you can already capture a very similar signal compared to using the deep learning-based score. And after removing the variants that are overlapping the coding sequence and the canonical splice sites, which are known to harbor existing autism associations, then even if the brain signals are still among the top, the relationship between the DIS and the local GC content is still very clear, as showing the plot on the right. And after removing these variants, none of the tissues are significant after after performing FDR-based correction, but the brain tissues are still marginally significant. Next, we try to directly correct for the effects of the local GC content on the DIS. That is, we regress out for each variant, we regress out the local GC content value from its deep learning based score and repeated this analysis. And what we saw right here is that not only none of the tissues are significant after multiple testing correction, you also don't see a prominence of brain-related tissues compared to the other GTX tissues. So the conclusion here is that when we use the local GC content alone instead of this entire deep learning-based pipeline, we could already capture a very similar association signal. In other words, perhaps you don't really actually need a deep learning framework to identify this brain-related signal, or you can think of it as this way: the deep learning-based score is more or less kind of like a Rube Goldberg machine in this case. But then the remaining question is, now that we have already established that the deep learning-based signal can be explained by local GC content, then where did this local GC content signal itself come from? Since autism is a disease that is more prevalent in males than in females, we looked at the sex combinations of the samples in the Safari SSC cohort. Among these families, you can see that the majority of them are having male probands, and there's about half of them are having male siblings, and the other half are having female siblings. Here, we performed a comparison of the local GC content between male proband male sibling pairs and the male proband female sibling pairs. And we observed that this brain-related signal is mainly coming from those pairs where the proband is a male and the sibling is a female. We also try to stratify the variants based on their relative orientation with respect to their nearest TSS, because the upstream and downstream TSS may be implicating different transcriptional processes, such as upstream for promoters and downstream for gene bodies. Here, we conducted the same analysis, but we divided the variants based on their orientation with respect to the TSS. That is, for male proband female sibling variants upstream less than 100 kilobases upstream of TSS, and for the same variants that are less than 100 kb downstream of TSS. And the main observation here is that the brain-related signal is mainly coming from those that are upstream of their nearest TSS. But then again, even after all of these analyses to narrow down the source of the signal, we noticed that there are still two main limitations of the analytical procedure that we've been using. The first limitation is with respect to the scope of the gene expression defined gene sets. Right now, the gene sets here are defined based on the expression across the different GTEx tissues. And we think we could include more gene expression data sets and have a more refined way of defining these sets. And the second limitation is that right now, this analysis only evaluates on the local GC content difference between the proband and sibling variants. And we think that there could be some additional sequence signal beyond the local GC content, maybe in the high dimensional sequence space. So to answer these two questions, we proposed this a more generalized framework, which is inspired by the previous analytical procedure that integrates both gene expression and the sequence to understand the association between autism and normal variants. First, to create these more fine-grained gene sets, we utilize this GeneShot resource, which compiles tens of thousands of different RNA-seq experiments and have computed this gene-gene co-expression correlation matrix. From this correlation matrix, for each gene, we can define a neighborhood of variants that is just the top n variants assigned to the nearest neighbors of these genes based on the correlations. And after forming each of these gene neighborhoods on these variants, we can first identify the GC content differences between the proband and siblings, and also conduct some machine learning analysis in the k-mer stage to see if the k-mers or the sequence context can be used to distinguish between the autism proband and also the siblings. Here, for each of the neighborhoods, we conducted a Mann-Whitney U test on their local GC content differences, and after a Bonferroni-based multiple testing correction, we identified 12 top neighborhoods, which are showing the most significant GC content differences. And based on these neighborhoods, we also conducted a goal enrichment analysis, where we compared the genes in these neighborhoods against the background set of genes, which is the superset of all genes that are being overexpressed in the GTEx brain tissues. And we found that even with this GTEx brain genes as background, we are still seeing these synaptic-related goal terms being significantly enriched, which validates our usage of the GeneShot expression as an additional resource for defining more fine-grained gene sets. And these goal enrichments also mirror the previous findings implicated by the ASD genes. In the next step, we're trying to see if there's any additional sequence signal that is being unexplained by the local GC content. So for each of these neighborhoods, we split it randomly into training set and testing sets. So on the training set of variants, for each variant we counted its spectrum of k-mers into this table right here, and then we just use a multinomial naive Bayes classifier to try to see if it can distinguish between the proband and sibling variants based on their counts of the k-mers. And after we train this classifier, we apply it to the other variants in the testing set, and then we compare to see if its performance is better than both the local GC content and the DI score. And the train test split is being repeated for 20,000 times to account for the randomness in terms of the splitting. Here in the left figure, we're showing the relative performance change when we use the different lengths of k-mers. You can see that when we increase the k-mer length from 1 to 7, the k-mer of length 6 is showing some sort of improvement when compared to just using the 1-mers, which is basically the same as local GC content. And when we applied the k-mers of length 6 to the top neighborhoods, we see that in each of these top neighborhoods, in most of these top neighborhoods, then using the naive Bayes classifier and 6-mers can have a better performance than just looking at the local GC content. But overall, right now, despite the fact that the naive Bayes classifier is outperforming the local GC content itself in this case, the next step would be to actually extract the relevant k-mers that are driving these predictions, which is more challenging. So overall, we revisited a previously found ASD association signal using the proposed deep learning-based model. But here, in our analysis, we show that a very similar association signal can be captured using a simpler sequence metric, that is the local GC content. And we further narrowed down the source of the local GC content association, and we saw that those are being driven by the de novo variants from male proband and female sibling pairs that were less than 100 kilobases upstream of the nearest TRSS. And then we also proposed a generalized framework that tries to integrate both expression and sequence context to investigate this disease association. Yeah, I would like to thank Jason Ernst Lab and also the NIH for providing funding.
Any questions?
Could you, I think I don't quite understand why the brain, so the scatter plots early on, it's
which one are you referring to?
Earlier in the talk when you're showing the Mann-Whitney p-values.
So here?
The dots.
This one?
Yeah, so here, it's gene sets defined by GTEx tissues, and you're looking for de novo variants that are within a certain distance of those genes?
Yes, the de novo variants are being assigned based on the distance to the tissue.
And so do the brain genes emerge because they have different GC content than genes from other cell types?
Well, the basic idea of this plot is that the probands and siblings are having greater GC content difference when they're near the brain genes.
It could also be that brain genes in general have greater GC content than the promoters.
But how would you explain the sex difference? Here. Yeah, keep in mind it's only being observed for the male probands versus the female siblings. And also keep in mind that this type of simplex-based study design, it's already trying its best to eliminate most of the technical confounding artifacts because the proband and siblings are presumably being sequenced together.
This aspect is this telling you that males and females have mutations interact with GC content differently in males and females?
Oh yes, I think that could be one of the takeaways.